Welcome to this video on protein sequencing via endopeptidase digestion. My name is Jonathan Chan. I am an incoming fourth year medical student at the UP College of Medicine, and I'm making this video because I haven't really seen a video that tackles on this particular topic in depth. So this first video is going to be on the basics of protein sequencing with this form of questioning with endopeptidases. The second one would be on this harder question, which I really haven't seen a video on. But I want to preface this video series, like this two-part video series, with the basics, right? To have a firm understanding of what really happens with these kinds of questions. So first things first, let's try to know what endopeptidases are, right? So endopeptidases from the word itself, okay? So endo, meaning inside, peptidase, so you have a suffix here, ase. This just refers to the fact that there's an enzyme. So you have cleavage within the peptide bond, right? So peptide bonds are the ones that make up your proteins. So essentially, the point here is you want to find the correct sequence of your protein via digesting your peptide bonds. So let's take a look at the question. So it says here, determine the sequence of a nonapeptide. Nonapeptide meaning there are 9 amino acids, right? So with amino acids given over here, if treatment with the following reagents and peptidases result in the following. So you're given a bunch of reagents here in peptidases. So trypsin, chymotrypsin, pepsin. These are endopeptidases. Your cyanobromide is a type of reagent that's used in protein sequencing, and it's very specific for an amino acid that we will see. Okay. Now, to illustrate what happens here, I just have a picture of a peptide bond. So as you can see over here, we have the N terminus or the N terminal and the C terminus or the C terminal. Now, what cyanobromide does actually is it cleaves, right? It cleaves methionine, M, that's its specificity, at the C terminus. At the C terminus. Okay, what would that imply? So if you have methionine over here, you're going to cut it at this point. This is called a sessile bond. So if you have methionine, it's going to cut off at the right side. This right side is the C terminus, as you mentioned, right? The C terminus, so it cleaves off over here. Okay. Later on, which is the N terminus, okay, we're going to see that. Like let's say if it were N terminus instead of C terminus, you have methionine over here, it's going to cleave off at this position. So it's at the left hand side. Okay. But this is very specific. It cleaves methionine at the C terminus. For trypsin, what we're going to see is that trypsin actually is very similar to cyanobromide. It just has a different specificity. In particular, it cleaves off lysine and arginine also at the C terminus. So just be familiar with the different one-letter symbols for these amino acids. Like lysine is K, arginine is R. Okay? Now we take a look at chymotrypsin. Chymotrypsin is also similar in that it cleaves off also at the C terminus, but it just has a different specificity of amino acid. In particular, we have phenylalanine, tyrosine, and tryptophan, right? So they're still all at the C terminus. The biggest difference would be at pepsin. So when you treat a peptide with pepsin, it's going to cut, instead of the C terminus, it's going to cut at the N terminus. It's very similar to chymotrypsin in its specificity in that it includes phenylalanine, tyrosine, tryptophan. The only additional amino acid specificity would be the one for leucine. So these are your rules. So using these rules, we can actually determine the sequence of an amino acid. So how is this done? So let's see, a sequence of a polypeptide, yeah, with those amino acids. So how is this done? You first draw 9 blanks because there's going to be 9 different residues present in your peptide. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So we'll label them as such. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so let's start with cyanobromide. So for cyanobromide, it's going to cleave methionine at the C terminus. And you would notice that you're forming a pentapeptide and a tetrapeptide. So, since it cleaves methionine at the C terminus, that would imply, of course, that methionine should be at position number 5. Right? So why is it at position number 5? Because if it cleaves over here, you're going to form a pentapeptide and you're going to form a tetrapeptide. Right? No other position would give you this kind of pattern where you have a pentapeptide and a tetrapeptide. So we're sure that this is the position of methionine. The others you're not sure yet, okay, because it's only cyanobromide. So we're going to use a different endopeptidases to get their position. So we're just going to copy, okay, these aren't set in stone yet. So G, E, F, and K. And here we have L, A, R, C. Okay, next we proceed with trypsin. So for trypsin, you would notice that it would cleave off at K, lysine, and R, arginine, right? Forming a dipeptide, okay, another dipeptide here, and this pentapeptide. Okay, so what would this mean for our sequence? You see here, you have lysine, right? And if it cleaves off over here, you're going to get a tetrapeptide. But you see that you actually formed a dipeptide over here. So that would mean that K, lysine, should be over here because it's going to give you a dipeptide, right? So that would necessarily mean that K should be over here. Moreover, E or glutamic acid should be over here. Why? Because if you cleave it over here, there's actually nowhere else for the E, amino acid, glutamic acid to be in. So you're sure that they're there, right? You're sure that they're there. You don't know whether, you've used up E and K, but you still don't have F and G over here. It's still unknown, right? It can either be FG or GF. We still have that to be determined. We already know what methionine is, so that we're gonna leave it at that, right? So methionine is present there. We know this is its correct position. How about arginine? So arginine should give you a dipeptide. It can't be over here because this is arginine. If you cut it off over here, right, we're going to form a monopeptide. So this would mean that arginine should be over here because if you cut arginine there, you're going to form a dipeptide, right? Therefore, the position of arginine should be over here, and it gives us La, right? So we are going to put that over here, okay? And there's actually only one place where you can put cysteine. Cysteine is only going to be over here, right? So this would mean that we know where arginine is and where cysteine is. What we're not sure of would be the leucine and alanine, because leucine and alanine can also very well be Al instead of La. So we're going to use chymotrypsin and Pepsin X. So we've already done cyanobromide and we've done trypsin. Now let's take a look at chymotrypsin. So for chymotrypsin, it's going to cleave phenylalanine, tyrosine, and tryptophan at the C terminus. But here we only have F, right? We only have phenylalanine. So for phenylalanine, F over here, it's going to form a tetrapeptide. So if it's going to form a tetrapeptide, it can't be over there, right? It has to be over here. Why does it have to be over there? Because if you cleave it here, you're going to form a tetrapeptide. Therefore, we know that phenylalanine should be over here and glycine should be over here, right? And this is already sure, right? Because we've already used the rules for cyanobromide and chymotrypsin, and they turn out to be correct. We still don't know whether this is La or Al. Why? Because when we cleaved off here, right, for chymotrypsin, okay, we're still left with pentapeptide, right? MCRLA, which is essentially this, right? It's still the same thing, just in a different arrangement. So now we're only looking at LA. Is it LA or AL? Okay, so that's where pepsin comes in. Notice that in pepsin, we have AL, right? So, leucine would be cleaved off at the N terminus. What does that imply for our problem? Uh, it can only be here, right? Because it will cleave off at the N terminus or the left side of leucine. Because if you have AL instead, what's going to happen is you're going to cleave off here and you're going to have a monopeptide. But as you can see here, you actually formed a dipeptide. Therefore, the correct position is LA. Right? So we've now determined what the correct sequence of our amino acids are in this polypeptide. It's EKGFMCRLA. Okay? So that's how you solve this kind of problem. In the next video, we're going to be talking about this next problem, which is slightly harder, and I haven't really seen an example of on a YouTube video. So stay tuned for that.